I'm your host. Okay, let's start over. My name is Jianqiang. I'm your host today. So I'm a postdoc at UC Berkeley working with Peggy Lemmer. Today's workshop brings together complementary perspective on LC-MS-based proteomics to study protein interactions and post-translation modification in plants. We will work through the workflow from experimental design to data interpretation. We are delighted to have Dr. Ling Shi and Dr. Zhili with us today, who will share their expertise and perspective case study. Together, their talks will highlight key considerations for interactome and post-translational modification study, as well as how experimental choice influence downstream and analysis and biological data interpretation. Please feel free to put your question in chat channel through the talks. We also welcome follow-up discussions, consultation, and potential collaborations after the workshop. Today's workshop was co-organized by Plant Postdocs and powered by NASC. So the first speaker is Ling Shi. She is a postdoc researcher at the University of Hohenheim, focusing on quantitative proteomics, particularly doing interactome mapping of membrane-associated proteins. She will introduce APMS and Turbo ID workflow for studying membrane protein interactomes with practical insight into experimental design. Let's welcome her. I will stop share. I will let you share. Ling, stage is yours. You were muted.
Okay, so now can you hear me? Yeah. Okay, great. And I hope you can see my screen,
Yeah,
right?
Ling, you are in the presenter view.
Presenter view.
Yeah. If you share the other screen, it will work.
Okay. Let me redo it again. There are technical issues as always. Share my screen. And then
Yeah, that's cool.
yes.
Can you show your slide?
Now?
Yes, that's good now. Thank you.
Okay, great. Okay, sorry for a little bit hiccup. And thank you very much for the nice introductions. And I'm going to talk about a little bit about my experience working with mass spectrometry for detecting protein-protein interactions. To start with, and we have the first question, what do we know about protein-protein interactions? So here I take an example from the plasma membrane protein. So typically, and we see the receptor-like kinase, for example, the BRY1 and BAC1, and we can see that their interaction is induced by the brassinosterol at the plasma membrane. So in this case, we know that for the protein-protein interaction, actually these interaction can be induced. So secondly, we can see that the protein-protein interaction is not always there, it is transient. So as soon as the signaling can transduct from the extracellular domain into the intracellular, and this protein complex can be dissociated at some certain domains. And also, we know that the protein-protein interaction is always coupling with post-translational modification in this case. And again, another feature we know that for protein-protein interactions, it is always not one-to-one relationship. Sometimes it's one-to-multi relationships. For example, here, one of the receptor-like kinase extracellular domain circ1, and it has two different patches, and based on the structure complementary mechanism can interact with two different type of the ligand and along with their correspondence receptor-like kinases. Okay. So far, we know that protein-protein interaction is a complex process and they have certain dynamics. So the nature of the bonds between the two big molecules for proteins are non-covalent bonds. So normally we know that these bonds are referring to a short-range interactions and typically van der Waals interactions, hydrogen bonds, or the salt bridge. And one of the things why we need to know about this nature of these bonds, and for example, in our protocols, if we're using different kinds of buffers, and if these buffers can interfere with these bonds and can potentially interfere with the protein interactions, and which will give us a different results. So why mass spectrometry is needed in protein-protein interaction studies? So firstly, the benefit is we can learn this phenomenon in vivo. And secondly, because mass spectrometry can do the quantitative information. So in this case, it can provide you a certain pattern to describe the dynamics for the protein-protein interactions. And also, mass spectrometry has ability to distinguish protein families. So if you have a very complex samples, and you can distinguish your proteins from these complex preparations. So nowadays, there are a lot of different methods you can use, which is based on mass spectrometry. However, not all of them are suitable in the plant science field. And so far, the most two popular one is affinity purification mass spectrometry and also the proximal labeling mass spectrometry.
Okay.
So, all right. So then I would like to compare a little bit about these character within these two methods. And one of the character is the interaction radius. And we can see that for the affinity purification mass spectrometry, or sometimes we are referring to affinity enrichment mass spectrometry, the interaction radius is ranging from 3 to 10 nanometers. And for the proximal labeling mass spectrometry is ranging a little bit further from between 5 and 15 nanometers. And also the two methods have different biochemistry basic. And for the APMS, it's depending on the antibody affinity on the towards that certain tag. And for PLMS, it is actually influenced highly by the bioaccumulation efficiency. And based on these principles, the type of interactions that they can capture is slightly different. And in the APMS, normally they're more suitable to capture the stable, transient, partially direct, or indirect interactors. And comparing with that, proximal labeling mass spectrometry, and normally it is suitable for capturing partially proximal interactors. And based on these principles, of course, you can imagine what can be the contaminations in the end. For APMS, it's a non-specific binder is normally composed to the contaminations, and for PLMS, the non-specifically labeled proteins is composed to the contaminations. These informations are important for directing us to choose different type of controls in the end. So I would like to briefly introduce you the workflow of the mass spectrometry-based interactome experiments. And more or less the same, I use an example from the AEMS. And in this case, there are 4 stages to carry out the whole workflow. And the first stages, of course, it is protein extraction steps. And the second stage is biochemistry step. And here it is AEMS example. So it is a single step AE, meaning that using a single step pull down and using this antibody to pull the bait protein along with their interactome out of the complex sample, and then to do a mild wash to keep as much as the weak interactors as possible in your samples, and afterwards to elute via trypsin digestion into the peptide form. And of course, the third stage is doing a mass spectrometry analysis, and the fourth stage is carried out the data analysis. So for the mass spectrometry quantification methods, so normally we are using label-free quantification, but it doesn't rule out the other possibility. You can choose the way that you work the best in your lab. And for a lot of labs who doesn't have a mass spectrometer directly, I assume you will send your samples to your collaborators or to the mass spectrometry centers. So I would later to focus on the first stage and also the last stage. So this is something that you may need to handling by yourself. All right, so let's have a look firstly at the APMS sample preparations. So basically there are 4 steps. Of course, the first step is create your own construct. You have a protein of your interest, then you need to select a certain tag. So GFP is quite popular because it's fluorescent protein and strongly expressed, and you can always monitor how strong your protein expressed and where it is pressed. But of course, you can also choose the other tags, for example, flag tag or strep tag. So then afterwards, you would transform this tag into the plant organism. So transient expression in tobacco or to stable expressed inside of Arabidopsis. So at this stage, and there should be, or there requires a certain experiment design and which control to use in this case. So normally, the mock control or wild type is always welcome. And if you are using GFP tag, it is highly, highly recommended. Please take an empty GFP vector as a control as well, because GFP is a relatively larger protein, so it can get very sticky inside of the samples. And another thing is quite special, and depending on your biological questions, so if you would like to have a look different treatments effects on the interactomes. So in this case, you can use exactly the same material, but treat it with a control condition. So this can also work. All right, so as long as you have designed your experiment, prepared your sample, then you do lysis. So normally you will lysis using a total protein extraction protocol, or if you have a specifically interest at some certain organelles of proteins, you can do a fraction enrichment, for example, memory fraction enrichment. And the first step, as long as you have your proteins ready, and then it is biochemistry, do the pull down using the anti-GFP beads. So comparing with the APMS, then we go to the PLMS protocols or PLMS steps. So more or less the same. Still, you need to create your own constructs and pay attention to the tag position, N terminal or C terminal as usual, the construct issues. And normally, or the publications cases, and they always like to also include a fluorescent protein inside of a turbo ID construct. So in this case, again, it is yourself to monitor where your protein express and how well your protein express in your system. And for the experiment design, the control of choice, again, normally the mock or wild type is mostly welcome, and please take them into it every time. And one of the special thing in turbo ID experiment design, and you can also choose an unrelated protein as a control. This is highly, highly depending on your scientific question. So I will explain it a little bit later in the data analysis processing. All right, so before you lysis your protein, and you have to do another step here in the PLMS protocol, it is biotinylation. And this is depending on the tissues or depending on the seedling ages. So the biotin concentration or the incubation time is quite varies from minutes to hours. So there has been a lot of really, really good protocol has been published about these numbers. So it is recommended that you can optimize your own protocol based on your question, based on your tissues, and based on your experiment design. And again, for the last step is doing the pull down. And one of the things I would like to emphasize here is before doing a pull down needs to remove the free biotin from your protein sample in this case. So then we come to the part that's talking about a little bit about data processing. So whether if there is a specifically tools for data processing, and from the publication or from our experience, there's no specific really powerful tools for such a thing. So I would like to share a list of the things that you can use if you start to do this work by yourself. The first one is very common one, using an Excel table to open your data, and then you can view your annotation about your protein information here. And if your data is too large, sometimes you cannot really use Excel to open it. Also, you can use Python or R to open it and to process it. And also in Python R, nowadays they have a really, really good bioinformatic packages, and you can download it for free. And to learn it is quite easy as well. So for persons who doesn't like to code, and also there is a recommendation The software is called Perseus. It was developed by MPI. It is actually a traditional software that you can analyze the proteomics data. Also, the one that is very user-friendly and also doesn't require much of the coding skills for data visualization is called Orange. And of course, if you're good at Python, you can also use this data visualization tool to combining with a Python coding as well. And for nowadays, I think you already know that there is a lot of AI tools as well. So put it in a word that every method leads to the Rome. And then I would like to share you the very, very general steps for data processing to get your Interactome ready for you. And again, there is no only one way to do it, but I would like just to show you some general points, and also depending on your collaborators and depending on your mass spectrometers, so some details of the steps can be varied. So the first step is cleaning the data. So now I use at the right side, there is a triangle numeric example to see how many protein groups in the matrix can be reduced in the end after a few steps of cleanup. And the first step is clean the data. So this is this is for the contaminations. These contaminations normally coming from the operators, for example, the skin cells or the salivas, so they can go into your mass spectrometry data in the end. We have to exclude it. Also some unspecific signals from the machines, and we have to exclude it as well. So in the numeric, what we can see original, maybe we have the matrix about 2,700 protein groups. And so after the clean data, so the matrix is slightly reduced. Then the second step is quality control. It's a term nowadays we say it's QC step. So quality control is really, really important for analyzing the data, the Interactome analysis. And again, there is not only one way or one software to do it. So I would like only to show you a very, very simple two ideas how to do the QC. So normally we would like the replications to be reproducible. So sometimes we have really, really bad data generated. We have to exclude it. So one of the way is using the pairwise Pearson correlation matrix. So in this case, you can set a threshold using the R-square values. If the threshold is below the certain R-square values, and you have to exclude such replications. And another way to viewing it is using histogram distributions. And normally, or they should be theoretically, and your mass spectrometry data, when you plot the counts against the abundance, and they should be a normalized distribution. So if the normality of such a distribution has been heavily disturbed, and this data is a bad data, so you have to exclude it. All right, so after the data exclusion from the numeric example from the far right, and we can see that the matrix has been further reduced. And then of course, we can go to the third step, but before we dived into the third step, so one of the things I would like to mention that we normally would like to check very important information. This is BATE, or the protein of interest abundance, relative to the full data distribution. So here I present to you and two examples from TurboID and AEMS. So on this this viewing plot actually is coming from a TurboID data set. And a protein of my interest, we can see that it has relatively high abundance on these distributions. And this is very makes sense because the protein of your interest is the closest relationship towards a biotin ligase. And if we wanted to use a cytosolic TurboID protein as a control, and there is a trade-off effect can happen. Why? Because in this case, sometimes we also see that the protein of our interest has a really high abundance to pull down in the cytosolic samples, meaning that cytosolic control can get really close to the protein of your interest. So in this case, the trade-off effect can be in order to get rid of the cytosolic interactor as possible, you will lose some of your interesting interactions as well. So this is based on the biological question, which way you want more or which way you want less. So comparing with that in the mock control, and we can see the data volume is heavily reduced. And also the protein of my interest is significantly lower than the other two groups. Similarly, if we're looking at the AEMS data, we can see that in the AEMS conditions, and we can see the beta protein abundance is stay at the very high rank comparing with the wild type. And in this case, if the scientific question is I wanted to having a look my receptor-like kinase interactors, so then I would use the wild type as a control and using one of the condition as the treatment. However, there is one case you may need to think a little bit more. If you wanted to comparing with different conditions in this case, for example, condition B and condition C, and we can see that there is a difference between the beta protein levels identification here. And maybe, or you might think to use the abundance of the beta protein as a normalization factor before you dive into the statistics. All right. Then applying data statistics, comparing with controls, and build the thresholds for your own interactome. So here, again, it's not only one way to do it, but I would like to present to you three aspects to view your data in this case. So firstly, you can choose to use the fold change in this case. So of course, taking advantage of the normality distributions of the data, and then the threshold is already shown on the graph based on the percentage of the differences between the treatment and the control. And another aspect is using the statistic threshold, and normally it is volcano plot. This is quite common to see in the publications, and depending on the volume of your final data, this threshold can be varied. But one of the things, typical character is that protein of your interest, normally they're located at the far right corner. And the third aspect is specific binders. So in this case, and you would look for only the ones that identified in the protein of interest biomes, but not in the controls biomes. So this can, again, based on your scientific question, this list can be very valuable in the end sometimes. So, okay, right. So with that, so these are the general protocols I would like to share. I hope that I already prompted you some informatic tips, so help you to working through your own data. So now I will take only a few minutes more to show you an example that how we apply this interactome to study the dynamic of our interactions. So one of the examples that is we wanted to learn, we had a specific scientific questions. So here, originally, we found heterodimers is called SERC1 and QSK1, and both of them can form a heterodimer under sucrose treatment. And in this case, the downstream target is aquaporin. And of course, this process is highly dependent on the SERC1 kinase. And then the question naturally was coming, and what is a ligand? And we had a whole list of the different peptides. And in the end, we have high score peptide is called PEP7. So one of the evidence we wanted to collect is we ask ourselves, can we use the AEMS to help us to understand whether if PEP7 can be a ligand candidate? So in this case, we design our experiment in this way. We use the SERC1 GLP lines, and then we apply 3 different types of treatment, the mock control, the sucrose treatment, and PEP7 treatment. And in this case, we actually extract the microsomal fractions and then do a GLP pull down. And in the end, we analyze this data just comparing with the treatments. All right, so here is the data. So in the end, we find the downstream target of SERC1, like QSKs and also aquaporins, actually, they have really, really high correlation between the PEP7 treatment and sucrose treatment. And so in this case, it is a piece of strong evidence to tell us, okay, PEP7 is a really good hint. And furthermore, we use FLIM experiment can confirm that QSK1 GFP fluorescence lifetime is heavily decreased during the PEP7 treatment along with the SERC1. So this is one of the example. So furthermore, we ask ourselves, can we really use affinity enrichment mass spectrometry to capture the signaling crosstalk at the plasma membrane? So in this case, we designed an experiment that we use different treatments and we try to collect the interactome for SERC1 receptor-like kinase, also for BRY1 receptor-like kinase. In this case, we saw that on the plasma membrane interactors, they are heavily overlapped. Then we go to the database to do the data retrieving. We found the specific substrates for BRY1 and specific substrate for SERC1, and they also are heavily overlapped. And again, we would like to construct the topology for between these two receptor-like kinase. So we use the kinase target relationship. And these published data has showed us a directional network at the plasma membrane between these two receptor-like kinases. And so go back to the interactome, we have the induced interactomes via brassinosterol or PEP7, and then we're mapping these interactors directly on topology of this directional network, and we see that the topology features are heavily overlapped. So in this case, we had a hypothesis. Probably the brassinosterol is disturbing or is activating somehow the SERC1 and PEP7 signaling in the same line. And furthermore, in vivoli, we really detected that the PEP7 level can be levitated by brassinosterol application and can be inhibited by the inhibitor. All right, so that's my examples. Short summary. So I hope I convince you that, or I show you that both of the interactome analysis method are robust and powerful. And again, based on your biological question, they also require complementary methods to co-validate the conclusions. And one of the things that we care mainly about, it is what is control to truth. Again, to emphasize that depending on your experiment requirement, depending on your scientific question, also please keep in mind considering the trade-off effects if you choose a specific control. Other considerations, for example, plant materials or bait protein character or biochemistry system, for example, even the bead producer. And one of the small tip, please do not choose different bead producer to compare without their affinity. And sometimes it will give you a very confusing results. All right, with that, the last slide, I would just to briefly thanks to all the colleagues and friends, and that I only listed the person who has a contribution in this presentation. And also thank you all for listening, and I'm happy to take questions after it.
Yeah, we probably won't have time for this talk, but we will ask a question by the end. And okay, I want to give enough time to the second speaker.
All right, then I give the stage to Zhi.
Yeah, and let me introduce Zhi Li. And he is a postdoc researcher at Iowa State, working with post-translational modification-mediated regulation of plant signaling with expertise in phosphoproteomics and LC-MS-based multi-omics. Today, he will discuss the post-translation modification-focused LC-MS workflow and the key consideration for sleeping protein regulation in plants. Let's welcome him.
Okay, thank you for sorry. Thanks for the kind introduction. And my name is Zhi. I'm a postdoc of GDCB from Iowa State University. And today I would like to share some experience of using LC-MS to study post-translational modification. For the so for proteins, they can be modified in many different ways. In fact, there are more than 400 types of post-translational modifications have been described across biology. But not all PTMs are equally common, and even fewer are experimentally accessible. So the funnel on the left shows that a key point for this workshop as we move from biological reality to what LC-MS can actually measure, the PTM space narrows very quickly. And at the bottom are the PTMs most often studied by LC-MS. Their phosphorylation, acetylation, ubiquitination, and methylation. They are often studied is not because they are the only important ones. They are often studied is because they are the most treatable ones. So PTMs are not just decorations on proteins. They fundamentally change how proteins behave. PTMs can turn enzymes on or off, control protein stability, redirect proteins to different cellular compartments, and reshape protein-protein interaction. So because these changes do not require new protein synthesis, so PTMs allow cells to respond very quickly to signals. That's why PTM are so important to signal, stress responses, and dynamic regulation. Phosphorylation is one of the classic examples. It can activate or inhibit enzyme almost instantly. And ubiquitination is another classic example, often marks proteins for degradation, but it can also have signal roles instead of degradation. Glycosylation or liquidation can determine whether protein stays in the cytosol, goes to membrane, or is segregated. Acetylation and methylation frequently regulate binding interferences. And what makes PTMs more charming and challenging is that a key exception in modern PTM biology is a crosstalk between different PTMs. This means that one modification can influence whether another modification occurs or what its consequence will be. So instead of thinking about phosphorylation, ubiquitination, or acetylation in isolation, it's better to think of in terms of combined regulation. So there are reports shows that the phosphorylation can create docking sites for ubiquitin ligases, can linking the signaling to protein degradation. And also reports shows that acetylation can block phosphorylation by neutralizing charge or masking kinase recognition sites. Chromatin, which is a classic example where multiple PTMs work together as a regulatory code. So many PTMs have been studied using Western blot. So this Western blot is from me. One common approach is to infer PTMs from band shapes, where a modified protein migrates differently on a gel. But this can suggest that something was changed. However, it doesn't tell us what modification it is and where it occurs or how many sites are involved. So different motifs or even multiple PTMs can cause similar band shifts. So this approach is fundamentally ambiguous. Another widely used approach is to use antibody-based detection, such as phosphospecific or acido-specific antibodies. This method relies on entirely on the antibody specificity, and this can vary between antibodies. So cross-reactivity, epitope masking, and condensate can all affect what the antibody actually detects. But LC-MS allows us to directly detect the modified peptide, identify the exact modification site, distinguish between different PTM types, and quantify them globally. So instead of inferring PTMs indirectly, we can measure them directly. But that doesn't mean all PTMs are easy to say. So in a typical proteomics sample, modified peptides are outnumbered by unmodified peptides. For many PTMs, only a small fraction of protein population is actually modified. On top of that, many PTM peptides ionize less efficiently and produce weaker MS signals. So in certain acquisition mode, the MS spectrometer preferentially selects the most abundant ions for fragmentation. As a result, modified peptides are easily missed. That means PTM enrichment changes the composition of the sample before it ever reaches the mass spectrometer. So by selectively capturing modified peptides, enrichment increases their relatively abundance and makes them detectable. So this dramatically improves sensitivity, reproducibility, and site localization. On the right side are two common methods used for PTM enrichment. The upper cartoon shows that is the method we use to do phosphopeptide enrichment, and this is based on the theory of Lewis acid-based interaction. So the material we use to do the enrichment is iron or zirconium or titanium. And the lower cartoon here shows another method we use to enrich different PTM, for example, isolation or condensation or methylation. And this is based on the theory of immune recognition. So we can enrich those specific modifications with its antibodies. So PTM enrichment is not optional. It's what makes PTM proteomics possible. And here I would like to share some a protocol that used in our lab to do a plant sample preparation for LC-MS detection of PTM. And here is majorly focused on the phosphorylation. And you can find this article from the DOI number on the corner. I will go through from the very beginning here. So generally, the enrichment of PTM can be divided into two parts. The first part is the sample preparation, the peptide preparation for enrichment. And the second part is peptide enrichment. First, we need to grind plant tissues as fine as possible. The definition of fine, you can imagine matcha. So it should be as fine as a matcha powder. And then we add trace buffer phenol and also extraction buffer. And within the extraction buffer, we add phosphatase inhibitors. And I also read from some literature that for the extraction buffer, they also added the kinase inhibitor as well. But we didn't observe a difference with or without the kinase inhibitor. So it's up to you whether you want to add it or not. Then we separate the proteins after phase separation. We collect the phenol phase, and then you can repeat this process once to get an additional phenol phase and then combine them together. This is very important to increase your protein yield. And after protein precipitation with cold methyl and ammonium acetate, then you can get a protein pellet. But then you need to clean this pellet with 70% methanol for multiple times. So you add 70% of methanol in the pellet and then sonicate to recipient the pellet, and then keep this recipientation buffer in the -20 for half an hour, and then pass again and repeat this whole step for twice. This is how we get the protein as clean as possible. And then you can pass again to get the white pellet, but you need to dry it before you send it to the next step. However, you should dry it, but without overdry. If you overdry it, you can never use it again. So the key is to get rid of all those droplets, but make sure the pellet is as wet as possible. We usually use urea, 8 more urea to resolubilize the pellet, and we use a filter-aided sample preparation to again clean the protein to make sure it's as clean as possible. And then we do reduction and acylation with TCAP and IAA. From here, the IAA, the concentration of IAA should be used very carefully, because higher amount of IAA may cause some artificial modification, may cause some artificial PTMs to the protein. For our case, we usually use 10 micromole. You can adjust it according to your case. And then we'll perform unfiltered digestion. And here you also need to pay attention to the ratio between the enzymes and your proteins. So the enzyme we usually use is trypsin and lysine C. And the ratio is usually 100 to 1 white ratio. Because if you add 2 enzymes, you may contaminate your proteins with the peptides from the enzymes. After overnight digestion, we can connect the peptides. And then the peptides need to be cleaned and desorted with C18. So this process is kind of, well, it should be the easiest part of the whole process. Just be sure that you need to acidify your samples before you slow down to say in column, and then wash the peptides with multiple times of water, and then dilute it stepwise with a different percentage of acetylnitrile. Then you can dry peptides. Once you dry peptides, they can be stored at -80 for a long time. This is where you can take a break if you want. Then for the phosphopeptide enrichment, we usually use iron or titanium or zirconium in our lab. So, but we also observed the different enrichment methods show strong overlap, but also retain distinct and method-specific biases. So another option for us is to do a tandem enrichment. Now, by saying tandem enrichment, I mean, so in our lab, for tandem enrichment, we have two options. The first option, we enrich the phosphopeptides with iron, and then we keep the unbinding flow through, and then use titanium to do the phosphopeptide enrichment against this unbinding flow through. Then we analyze both fractions with MS. Second option, you can use titanium as the first step enrichment and do the column to enrich the phosphopeptides from the unbinding flow through. And a little bit introduction for the I want to introduce a little bit theory between different mass spectral oxidation mode. Once peptide enter the MS, there are two major strategies to acquire MS2 data. One is the data-dependent acquisition, or we call it DDA for short. And the other one is the data-independent acquisition, DIA for short. So in DDA, the instrument firstly performs an MS1 scan and then selects the most intense precursor science for fragmentation. So this works well for the abundant peptides, but low abundant species, including many PTM peptides, have been missed. So as a result, DDA can be a very powerful discovery tool, but it can be a little bit weaker compared to the DIA. For the DIA, they base it on the predefined windows. So this makes sure that across the whole scan, no peptides or empty ratios will be escaped for the scanning. So it captures everything and send them to MS2 for the identification. And here are some practical examples using our lab. We use both DDA and DIA for phosphoproteome study. But for the DDA, in order to increase the depth, we will use the help of tandem mass spec labeling. So a brief introduction for this. After we get clean peptides, we will firstly label the peptides with different tandem mass tags. And then we can combine this labeled peptides into one sample. Then you can do one step enrichment to get all those phosphopeptides. But for the DIA using phosphoproteome study, it can only do label free. That means we need to do phosphopeptide enrichment individually for each samples. So there's a basic comparison between DDA and DIA. For DDA, you will spend less time on sample preparation because you combine 18 samples within one and only do one phosphopeptide enrichment. But for here, you need to do 18 phosphopeptide enrichment. So less time for sample preparation in DDA mode. It also requires less cost on MS runs, but you need to spend a lot on the labeling kit, which is also very expensive. However, for DIA, it requires more time for sample preparation. Of course, it requires more MS runs, but you don't need to spend anything on the labeling kit. So this is a brief comparison. And here is some experiments, some data from myself. this is a phosphopeptide detected by DDA or DIA. So all these 6 samples are from Arabidopsis root proteins. And this is from a TMT label, DDA. From here, we can observe a large shared overlap across all 3 samples. But of course, you can always detect some individual phosphopeptides that only be observed in certain samples. For DIA, it also has a very large overlap shared by all 3 samples. But instead, the individual phosphopeptides can be found within each sample increased compared to DDA. For another case is that I'm interested in 2 proteins. I'm interested in the filtration of 2 proteins of myself, but I can only detect 2 phosphosites from the DDA sample. And then I switch to DIA, then I can increase this discovery into 6 phosphosites. And another one is that I couldn't find any phosphosites from a DDA mode. However, for the DIA, it gave me 6 phosphosites. And some of them are really interesting for me. So for your case, the chosen between DDA, DIA depends on what you want. So if you are just interested in how the global phosphoproteome responses to certain treatment, or whether knockout gene can affect the global phosphoprotein change, you can start with just want to get some general information in this case, then you can start with DDA. But if you want to do an in-depth phosphoproteome where you have a target phosphosite that cannot be or rarely detected by DDA, you may want to consider DIA. And here are some softwares that can be used for both mode-generated raw data interpretation. For DDA raw data, we usually use MassQuant. It is free. And for DIA, we can use DIAN. It's free as well. Another one is Spectronaut. You need to spend on it. And for the proteome discovery, it can be used for both DDA and the DIA mode, but it's also not free. And for dependency, it's required by different interpretation. For the DDA mode, owning a database with protein sequences is good enough for the search. For the DIA database, so for the DIA, there are 2 different interpretation modes. The first one we call library-free search. The other one is library-dependent. So for the library independent search, what you only need is a database like what you get from the DDA mode. However, for the library dependent, you require a database as well as a library. And this library is usually come from a raw data that generated on the DDA mode. And so for here, this DIA is generated by library-free mode, and this is what I'm sharing. What I'm doing right now is to build a good quality library, trying to increase the depth and also reduce the variance between different runs. And this is the workflow of data analysis after the raw data interpretation. So after the raw data interpretation, you will get a list of phosphopeptides as well as their quantities. So very similar as what Dr. Lin has just showed for the IPMS data analysis. So firstly, you need to remove contaminants because within the list, you can always find a lot of some proteins from like rats, humans, or the BSA may be involved in your buffer, and also peptides from the enzyme used for digestion if you add too much enzymes. Once you remove this contamination, you can do an imputation for missing values. And the way I use for my data is usually use the mean of other replicates or the median of other replicates for the imputation. And after that, I will do a normalization. For the normalization, I will choose total fraction or median. I don't think a very difference between the 2 different ways, but you can try both if you want. And then you need to do a quality control by using PCA to say whether there is any outlier. Then you may want to exclude it for the analysis. And also you can try intensity distribution of different samples, especially for those biological replicates, to say how stable they are. And my suggestion is to do a quality control before the normalization and after normalization, and then compare them. Then you can see how the normalization works for you. And after that, you can do some statistics to get the log fold change and the p-values, whatever you need for analysis. So to get an over image from your data, you can do a going range to say which pathways or biological process are involved or affected by your treatment or the knockout of certain genes. What kind of protocols, and then you can do a heat map, say pick up some specific peptides we're interested in to show how they behave. Motif. I think this is the only different thing that we can learn from this photoproteome data, because we know that certain motifs can be recognized by certain class of kinases or phosphatases, and this can be helpful to get more detailed information for your analysis. So for the software that can help as well, same. So Proteus, distributed by the Mascons group, and also R Python. Both are very powerful tools if you are familiar with coding. And these are two websites you can use for motif search and also for goal enrichment if you are not very good at coding. Some hints before you start your photoproteome study. Biological replicates is always the question being asked. So my recommendation is that you probably need to prepare 4 to 5 biological replicates for each sample. And since that for phosphoproteome, there is a kind of feedback. That means when the cell sends some signal, it may get phosphorated or dephosphorated. But after a certain time, sometimes maybe 10 minutes or half an hour, this change will become normal. So you can never detect anything after, let's say, 20 or 30 minutes. But this time depends on your target of protein. So I would suggest you to do a time course, and this may be helpful for your study. Another question is about how you tell the difference whether the phosphopeptide change is caused by the phosphorylation or by the decrease or increase of the protein itself. My suggestion is to prepare 2 sets. One for the PTM study, the other one you can do a global. Then you can get different information from both aspects. By comparing both, you can get a clear conclusion. And another thing is that I suggest you to use the tissue where you found the responses for the sample preparation. For example, I'm interested in root growth and how roots respond to different treatment. So I only collect the roots instead of the whole seedlings for my protein isolation. Because if you collect the whole seedlings instead of the tissue where you are really interested in, you may buffer some responses. So probably use the tissue, the specific tissue you're interested in is a good idea. And at the end, I would like to thank you for your listening, and thanks for the organizers for this kind invitation. And yeah.
Great. Thank you for 2 speakers. So we have 5 minutes for question Q&A. I've monitored the chat channel, so I've seen 1 question. I think for Lin, for interactome experiments of membrane-associated protein, are there any standard control protein used to distinguish non-specific binding? Lin, you can answer that question.
Right, I'm muted. Yeah, I just see this question. So this is really, really good questions that there are some synthetic proteins that people are constructed with a signal peptide to driving a certain protein at the plasma membrane, but not really something like you can use it. In my experience, not really something like you can use it and as a plasma membrane control, and you really can rule out non-specific binding. So sorry, I cannot provide you more information about it, but I remember that I heard somebody was someone had some synthetic signaling peptide to driving a certain protein at the plasma membrane to create such a control. Yeah, probably can look for it.
Okay, so the second question to Zhi is when you observe a change in post-translational modification signal, how do you distinguish between a change in the modification level versus a change in the total abundance of that protein?
Yeah, this is also being asked by a lot of reviewers, of course. So for my previous experience, because I used to work with short-term treatment like 3 minutes, 5 minutes. So this wouldn't be a problem for me. Currently, I'm intended treatment to half an hour and 1 hour, so it's become a real problem for me now. So my idea is to prepare 2 sets of peptides. One, just send it for the global search. The other one, you can do a PTM enrichment and then search for the PTM, then compare both, you can get an overview. So for the global search, you can make sure that your protein abundance will increase or decrease. And with that, you can make sure whether it's caused by the modification change is caused by the abundance change or not. So 2 sets can work.
Okay, so there is a question to Lin. Which detergent or stabilization strategy have you found the most compatible with downstream LCMS?
Yes. So firstly, mass spectrometry doesn't like detergent. I think Zhi is also agree with it. And the detergent for compatible and for the memory protein, yes, and sometimes you need some detergent to stabilize our proteins. And one of the recommendation is it's called DDM. It is called beta D maltosate. And this is a very, very mild detergent. We sometimes use 0.1% in our experiment. And in this case, can help you to dissolve your protein at the same time is not to retain in your mass spec sample. So if you need, you can choose something milder, same like this.
Yeah. Okay, thank you for your answer. I think our time's up. And thank you. Let's join me and to thank 2 speakers today. And thank you for all the audience for all your time and being here. And that's all for today.
Okay. Thank you very much.
Thank you.
Yeah.
Yeah. Bye-bye.
Okay. The speakers or the organizers can stay here. We can talk about
okay.
and